Bio 181, chapter 3, key concept is on proteins. So proteins have these very complex structures. They're held together by both hydrogen and covalent bonds, red. So both hydrogen and covalent bonds. So remember, this would be like that secondary structure. This is the tertiary structure, the third level of structure. And these can be disrupted by changes in pH, so a change in acidity, the acid-base concentration, and temperature. So both of these are really important to regulate for many organisms because a change can break these bonds, change the shape, and the shape of a protein is very important for its function. Another word for shape is conformation. Just means the same thing. The conformation of a protein is its shape. And if the conformation changes to the point where it's not functional, that means it's denatured. So it's denaturation. We've got over here, if you remember these, what do you think these are called? These spiral shapes. That's the alpha helix, part of the secondary structure. Right here, this would be more of the beta pleated sheet. Okay. And then if there are bonds between them, covalent bonds, those would be like a disulfide bridge. That would be covalent. There's also going to be other weaker bonds, which might just be between, if there's some R groups that are positive on the amino acid, they're going to interact with negative R groups, and that's going to pull it together. But as we change, so say we put a whole bunch of hydrogen ions here, this is a very acidic solution, then maybe this part of the protein is going to be attracted to the solution and it will break apart. So then you've got this protein, which you still have this initial primary structure here, but the shape has been lost, and the shape of a protein is very, very important to its function. So structure determines function. We have denaturation, we have a loss in function. So an example here is the fried egg. So the white of an egg is all protein, it's called albumin. And if you heat it up, so this was clear, right? So this part of the egg here, all of this was clear before, and then we added the temperature. That temperature broke apart the bonds, and it changed the structure of this protein, solidifying it and making it really non-functional in the way it was before. And again, that is called denaturation. And that is it for this. I'm not going to go into that. That's it for this video.